DNA is like the blueprint of your cells. It is what controls, it's what controls the development of any living organism. They are located within the nucleus, and this is an example of a strand of DNA. The DNA is often twisted, it's usually twisted in a double helix structure like this, and within the DNA are nucleotides. These nucleotides are made of 3 parts. The first part is the deoxyribose sugar, deoxyribose meaning without oxygen, and the phosphate, which is here, and a nitrogen base. There are 4 kinds of nitrogen bases. The first one is guanine, second cytosine, adenine, and thymine. Guanine will always be paired with cytosine, and adenine will always be paired with thymine. Now back to the deoxyribose sugar. There are 5 carbons. The bond that connects phosphate with the deoxyribose sugar is called a phosphodiester bond. The bond that connects guanine and cytosine and adenine and thymine are hydrogen bonds. Connected by hydrogen bonds, and this is important because hydrogen bonds are weaker and easier to break apart, and this is important when you're doing DNA replication to be able to split this apart, for the helicase to be able to split this apart. Cell replication happens during the interphase. The cell is growing and carrying out cell processes such as replicating DNA. It can't divide while replicating DNA, so it replicates the DNA before their division. Cell division is called mitosis. Okay. Enzymes are used in this process. They are used to speed up reactions and build up or break down items they act upon. The first enzyme that is used is helicase. Helicase starts at the start codon, which in DNA is TAC, and in mRNA is AUG. This is the start codon. This is what helicase looks for when it's going in and splitting the hydrogen bonds, thus separating the 2 strands of the DNA. Okay, if you're wondering, here is the start codon. It's TAC. Firstly, the enzyme helicase, I hope I spelled this right, helicase, which looks like this, will go in and separate the 2 strands of DNA. After helicase separates these hydrogen bonds, the second step to this process is that primase comes in and lays down a short RNA primer and DNA polymerase goes in and lays down new nucleotides according to the primer. So the third step is the DNA polymerase. Following the RNA primer, DNA polymerase will come in and replicate and lay down new nucleotides according to the primer. The third step, that's the fourth step, is for ligase. Is for ligase to go in and bind the lagging segments of the Okazaki fragments with the phosphodiester bonds. DNA polymerase, after laying these down, a different type of DNA polymerase will go in and check if this is correct. So it will check if the A is always matched with the T, the C is always matched with the G, and yeah. It also gets rid of the primer that primase put down. Ligase will go in and create phosphodiester bonds and eventually bind the DNA together, and that's what forms a new strand, another strand of DNA. Translation is when DNA, like we previously did, turns into RNA, transfers into RNA, and then into proteins. This process is prokaryotic. The next one we're moving on to is transcription. Transcription is when DNA this process is this means that it has a nucleus, and therefore it needs something smaller to pass through the nucleus's pores, because the DNA's double helix strand will not fit. Now, in protein synthesis, RNA polymerase breaks these bonds. Now, in mRNA, they're creating an mRNA because it needs to pass through the nucleus because it's small enough to fit through the nucleus's pores. Enzymes will come in and do the same thing as before and create the opposing nitrogen base. And because thymine is not part of mRNA, it's replaced with uracil. So instead, it goes G, and instead of putting down T beside A, it's now U, and this is G, and this is C. And it continues on like that.
After the mRNA is created, it is small enough to pass through the pores of the nucleus. After it is no longer in the nucleus, ribosome picks it up, and then tRNA will bring amino acids and form a chain of it on the ribosome, which are called polypeptides. This is the translation period. Now to talk about mutations. There are many different types of mutations. The first type is that if there's only a single nitrogen base that changes. Now, for example, for the code GAA, this code makes glutamine. And for example, if there's a mutation in the A and now at the G, the result of that would still be glutamine. But if, for example, this, which was original glutamine, changes, this A changes into a U, then this codes for something different. This mutation codes for aspartic acid. Now, see, if this mutation happens, nothing will happen to the final result, for the resulting proteins and amino acids. Whereas for this one, there is a change from this to this, and therefore, if the codon is different, then the amino acid and the protein would be different too, and this may cause a change in the organism's characteristics. Another example is if it accidentally codes, if the mutation causes it to code for a stop codon. Now, for example, originally if it was UAC, the result of that is supposed to be tyrosine. But if there's a mutation that happens in the C and it turns into a G, then this actually codes for the stop codon. The stop codon will stop the DNA polymerase from replicating the rest of the DNA. So, for example, if this is a DNA strand and you get that stop codon here, it would stop here, and the rest of this would not be replicated. Another type of mutation happens when a nucleotide is added into the sequence. If a codon was meant to be UAC, then GUA, creating tyrosine and valine, and the mutation causes a C to be added to the sequence. The codon is no longer UAC and then GUA, it is now UAC and CGU, which is now coding for tyrosine, and instead of valine, it's coding for arginine, and this A is being left out. This also causes for a different sequence and different proteins and amino acids, and therefore a different result, creating a different characteristic or a change in the characteristic of the end product. Or if UAC and CGU was supposed to be the original correct sequence, and the C was left out, and therefore created this sequence, and then this is also a change because a mutation caused the C to be left out. This changes the result from tyrosine to and arginine to tyrosine and valine. This is also a type of mutation. The last kind of mutation that could happen is one that can cause pretty drastic changes, because if this is the original code, they then add an entire codon into this. They each have their own individual amino acids. This is not only changing the amino acid and then therefore changing the protein, this is adding an entire new codon in, and therefore adding a new amino acid into the sequence. And this is likely to drastically change the protein result, and therefore the result of the living organism as a whole. These large changes are called frame shift mutations.